Hi, my name is Anthony Mustoe, and I'm an assistant professor of biochemistry at Baylor College of Medicine in Houston, Texas. I've had the incredible honor of being a member of the 2022 class of Beckman Young Investigators. I was also fortunate to be an Arnold O. Beckman postdoctoral fellow in 2015 when I was a trainee at the University of North Carolina. Together, this support from the Arnold and Mabel Beckman Foundation has had a profound impact on my career and on the work that my lab is now pursuing. Arnold Beckman was one of the great scientific inventors and entrepreneurs of the modern era. His inventions, including the pH meter and a spectrophotometer, revolutionized chemical and biological research by enabling scientists to measure previously invisible aspects of the chemical world. Dr. Beckman's career epitomizes how developing new measurement technologies can open entire new fields and have impact far beyond what the technologies were initially conceived for. This legacy of innovation continues to live on through the Arnold and Mabel Beckman Foundation, which Dr. Beckman created to support high risk, high reward research by early career scientists such as myself. Inspired in no small part by Dr. Beckman, my laboratory seeks to develop new technologies to measure how cells work, with the conviction that these technologies will enable new understanding of biology and unveil new opportunities for treating disease and engineering cellular systems. My lab is specifically focused on understanding how RNA molecules function. RNAs are one of the central classes of molecules in cells. They are commonly thought of as a messenger or mRNA that carries information from DNA about how to make proteins. However, RNAs also function as regulators, sensors, and scaffolds within the cell, and even can form large molecular machines that can do really remarkable chemical reactions. A key way that RNAs perform these functions is by folding into elaborate structures. The structure of an RNA can determine which proteins it binds, where it goes in the cell, its stability, and how efficiently it can be translated into a protein. The challenge is that RNA structures are very difficult to measure. Every RNA folds into a unique structure that is encoded by its particular sequence. These structures are microscopic and are often very dynamic. Notably, many RNAs do not fold into just one structure, rather they can switch between alternative structures. Thus, really what we need to do is to record multiple snapshots or movies of RNA as they populate their various structures. For the past decade, my research has focused on advancing a class of technologies called chemical probing experiments to measure RNA structures in living cells. In these experiments, we use special reagents that can enter cells and modify RNA depending on RNA's local structure. We then use sequencing to read out where those reactions occurred, and then computational modeling to infer both how the RNA was folded and its dynamics between potential multiple states. The Beckman Young Investigator Award gave my lab the freedom to pursue an extension of this technology. Could we use chemical probing to measure RNA structural dynamics not just in a test tube and not just averaged across millions of cells, but inside of an individual cell? This is an important problem because every cell is different. In the body, a neuron is obviously very different from a bone cell, but even cells that look identical on a laboratory dish can vary in important ways. For example, some cells may be stressed, some may be growing, and others are likely preparing to divide. We hypothesize that RNAs might change their structures depending on what's going on inside of an individual cell, and that those structural changes might be a key way through which RNAs regulate gene expression based on cellular needs at that moment. This required us to develop a new technology that can measure structural dynamics from minuscule amounts of RNA, with orders of magnitude higher sensitivity than what was currently possible. When we started, this was only an idea, and developing this technology proved substantially more challenging than we expected. Nonetheless, I'm excited to say that we are able to achieve our goal of making the first ever measurements of RNA structural dynamics at single cell resolution. Using this approach, we found that RNA structures do vary substantially from cell to cell. Even more excitingly, this variation is not random. It appears to be correlated with where cells are at in their cycles of cell growth and division, thereby revealing that RNAs can indeed function as structural switches that dynamically tune gene expression. This mechanism was only apparent when we are able to resolve structure at the single cell level. We think that this mechanism may be important in diseases such as cancer, where cell growth and division are dysregulated, and that we might be able to develop drugs that specifically target this switch. More broadly, our work suggests that cells may use RNA structural changes as a general way to tune biological function. Looking forward, my lab is continuing to build on these technologies to reveal where, when, and how RNAs unfold inside of cells, and how these structures drive cell biology. We hope these tools will help the broader scientific community discover many more RNA structural switches across biology, with applications ranging from understanding disease to engineering new biological systems. I would like to thank Josh Meehan and Lucas Kearns, the trainees in my lab who led this work, along with the many colleagues and mentors who helped make it possible. I am deeply grateful to the Arnold and Mabel Beckman Foundation for selecting me as a Beckman Young Investigator. This award gave us the freedom to pursue one of the riskiest and most exciting ideas we had when my lab was just getting started. Equally, if not more importantly, the lessons in mentorship, leadership, and the relationships that I've gained through my time as a BYI, and the inspiration to do innovative science to benefit society will live with me throughout my career.